Right, so back for part 2 of proteins. Click that to move it on. Okay, we'll look at the peptide bond. This is the link between amino acids and amino acids in proteins and peptides. There's the synthesis taking place. A couple of amino acids join together to produce a dipeptide, a peptide consisting of 2 amino acids. Formation of a peptide bond is an example of a synthesis reaction. Like many synthesis reactions, this reaction involves the loss of a water molecule. Conversely, breaking or hydrolysis of peptide bonds involves a gain of water. This is what happens in cooking and hydrolysis by enzymes during digestion and many other biological processes. The 3D character of the peptide bond gives rise to the twists and turns typical of peptide and protein molecules. If you click on the image, there's a web link which contains a lot more information on this. They're the designations of the 2 bonds. Again, the web link will explain what these are. Protein structure may be described in 4 levels. The primary structure is simply the linear sequence of the amino acids as the protein is synthesized. This really is just a long line of amino acids tied together by peptide bonds. It doesn't have the structure or function of a protein. However, forces like hydrogen bonding start to force the primary chain into local structures such as helices and coils. This process happens spontaneously, driven by the characteristics of different amino acids in the chain, with interactions with water being very important. So the protein structure becomes more interesting, but it still isn't a protein. It still won't do the job it is evolved to do. The overall tertiary structure arises from a combination of all of the secondary structures and often gives us a working protein. The diagram there is, you'll see these in biochemistry textbooks, and it is referred to as a cartoon. Now, if you want to know more about protein, what happens to proteins when we cook them, click on this video over here, egg frying. You can add a narration to it when you listen to it, and it'll give you a description of what happens to egg proteins in the reverse of the process we've been talking about here, so-called denaturation, where we go from the protein's normal shape, sometimes it's called its native configuration, to a denatured form. Well worth having a look at. Many proteins have a quaternary structure, that is, several proteins working together. Examples include hemoglobin, shown here again in a cartoon image, and polyphenol oxidase, the enzyme involved in non-enzymatic browning. So 4 levels of protein structure: primary, secondary, tertiary, quaternary, designated by 1 and a little degree sign through 4 and a little degree sign. For more background, see the link down here, which is where I got a lot of these images from, and that's a nice summary there of primary through to quaternary structure. Okay, so as well as nutrition, of course, proteins are very important in food structure, and there's much food technology interest in them. We'll look at various aspects of proteins and food structure. These are probably the main points of interest, though they do overlap somewhat. As mentioned in protein structure, they have extensive hydrogen bonding with water. This is just one example included to illustrate the point. There's no need to know this detail. Obviously, what we're seeing here is a water molecule with a hydrogen bond linking it to various amino acid residues, in this case glutamine and alanine over there, and again more water molecules. So this produces as I mentioned before, gives the rich 3-dimensional structure of proteins. These are 3 different foods which hold much of their texture and result in mouthfeel to their ability to hold water. Obviously, meat as whole meat, or meat as burgers, or a pastry are very different textures, and is associated with their capacity to hold water. So emulsions, as well as being integrated into protein structure and with other molecules such as sugars, proteins can be incorporated into large-scale structures, of which emulsions are probably the most important example. Using the word immiscible here, that refers to substances which do not form solutions like sugar and water when we mix them. They just form an intimate mixture, really. The continuous phase is usually the one present in the largest amounts, and this is a well-known food emulsion, mayonnaise, in this case a mixture of oils in water. There are a huge number of emulsions in food, some of which occur naturally and others of which are produced in food poisoning. Here's just a small number of examples. Emulsions can also be formed when a liquid or solid traps a gas, such as an air in a mousse or carbon dioxide in bread. So this is how to make an emulsion. You mix all in water and give them a really good shake. The mayonnaise formed is reasonably stable, but will eventually separate into oil and water again. As we can see, there are ways of helping stabilise the structure of emulsions, which are used extensively in the food industry. Here's another example of a formation of an emulsion. Beating an egg causes partial denaturation and produces a foam. That is an emulsion, trapping air. However, if the egg is overbeaten, the foam will collapse. And there we go, it collapses. Modern milk is highly homogenized. Historically, milk, which used to come in bottles, would separate with a lighter cream rising to the top of the bottle, a phenomenon of interest to many birds. We use very high-speed blending to homogenize milk these days. You use the word cavitation there. Cavitation is the formation of vapor cavities in a liquid, i.e. small liquid-free zones, bubbles or voids, and this greatly helps produce the homogenized milk, which when we buy it, doesn't separate unless you leave it for a very long time. It'll probably gone off by then anyway. Okay, moving on. Before we continue, some revision on the structure of fatty acids. They have a long carbon chain. I haven't shown all the carbons and hydrogens in it, but I'm sure you're aware, for example, that that's a CH3 group and that's a CH2 group. So this long carbon chain is hydrophobic. That is, it doesn't like water. They also have a hydrophilic head that does like water. In the notes that's got hydrophobic, it should read hydrophilic. So the fatty acid has parts that will dissolve in water and parts that won't. And conversely, it has parts that will dissolve in fats and parts that won't dissolve in fats. So this little image is a summary here of the fatty acid characteristics, and it makes the next few images a bit easier to see what's happening with. When we place a fatty acid in water, it self-organises such that the hydrophobic heads are embedded in water and the hydrophobic tails stick up in the air. This self-organisation is hugely important in biology in general, but also in food structure. Then self-organisation can take a big step further, with groups of fatty acids forming spherical structures called micelles, or Micelles, someone pronounces it. Here the parts that dislike water are hidden on the inside, and the parts that like water are on the outside. The basic structure is thought to be a possible origin for cell structures in life, which have a double layer very similar to this. It's also the basis of how soaps and detergents works. The oily residues get trapped inside the micelles and they're washed away. This is a micrograph of emulsion droplets in mayonnaise, and you can clearly see the micelles. We can stabilise the structure of food emulsions using stabilisers, and here's a couple of examples: egg yolk, lecithin, and sodium stearoyl lactylate. Note the fatty acid type structure of both. Okay, so folks, we will talk about the structure of proteins a little bit more when we talk about water and the structure of water later on. That's all for now on this.